Welcome students. Last episode we discussed the different forms of DNA. In this episode we will see the stability of a DNA structure and what are the factors which affects the DNA structure and how we'll manage the stability of DNA. First, the stability of the DNA double helix depend on a fine balance of interactions including hydrogen bond between bases and surrounding water molecules and base stacking interactions between adjacent bases. Slight variations in the DNA sequence can have profound implications on the stability of DNA duplex. For example, mutation in the base sequences that results from errors that occurs during DNA replication can result in mismatch that leads to the relative unstable duplexes. This instability is exploited by proofreading enzymes which recognize the mutation and replace it with the correct nucleotide. To gain an insight into DNA duplex stability and how it is affected by changes in the primary structure, scientists have studied the structure and thermodynamic stability of variety of DNA duplexes by using a combination of physical methods including X-ray crystallography, UV melting and NMR. First, the inherent stability of the DNA. The role of DNA as repository of genetic information depends on its inherent stability. The stability can be evaluated in terms of denaturation and non-enzymatic transformation. That is, very slow reactions can have a significant effect on DNA structure or function. First, a DNA denaturation. Denaturation can be induced by high temperature or change in pH. The process is not uniform and involves covalent bond remaining intact, hydrogen bond breaking, disturbing of base stacking. How the DNA is denatured by hydrogen bonding. The heterocyclic bases of single stranded DNA have polar amidino, gonadino and carbonyl groups that form a complex network of hydrogen bonds with the surrounding water molecules. Some of these bonds must be broken during duplex formation as the interbase hydrogen bonds are formed. The overall process is one of hydrogen bond exchange and the net change in the enthalpy upon duplex formation is partially due to hydrogen bonds formed or hydrogen bonds broken. For duplex of any significant length in this an exothermic process at ambient temperature, not surprisingly, the coming together of two large oleomeric molecules is entropically unfavored, that is, delta S is negative. Then denatures of base stacking. Interstrand hydrogen bonding is clearly important in driving the formation of DNA duplexes, but it is by no means the only contribution factor. In individual bases form stock stacking interaction which are major contributors to duplex stability, as base stacking is much more prevalent in duplexes than in single strand. Base stacking interactions are hydrophobic and electrostatic in nature and depend on the aromatically of the bases and their dipole moments. Base stacking interaction in nucleic acid duplexes are partially interstrand and partially intrastrand in nature. However, it is probably more informative to consider base pairs rather than individual bases as discrete units in order to visualize the stabilizing effect of base stacking. The degree of stabilization afforded by base stacking depends on the DNA sequences. Some combinations of base pairs form more stable interaction than others, so nearest neighbor base stacking interactions are important determinants of duplex stability. Base stacking interactions increase with increased salt concentration as high salt concentration masks the destabilizing charge repulsion between the two negatively charged phosphodiester backbones. DNA duplex stability therefore increase with increasing salt concentration. Divalent cations such as magnesium 2+ are more stabilizing than sodium ions, and some metal ions bind to specific loci of the DNA duplex. DNA renaturing. Renaturing is the reverse of denaturing, a rapid one-step process called annealing as long as part of double strand remains intact. If the double strand is not intact, Then the process in two steps, with the first being a relatively slow step in which complementary strand pairing occurs via random collisions. Thermal denaturing or denaturing through melting. DNA exists as double helix at physiological temperatures. The two DNA strands dissociate at elevated temperature. The strands reannealing when temperature is lowered. The reversible thermal denaturation and annealing forms basis for the polymerase chain reaction. DNA denaturation is commonly monitored by UV spectroscopy at 260 nanometer. What are the factors affecting the DNA denaturation? The midpoint of melting depends on DNA length. Longer DNA has higher temperature of melting point. The melting point depends on pH and ionic strength. High salt increases melting point, and then the melting point depends on the base composition, that is high Cg increases the melting point of the DNA. The AT rich regions melts first. DNA hybridization via renaturation. The ability of two complementary DNA strands to pair with one another as DNA hybrids reveals evolutionary relationships. The closer the evolutionary relationship between two species, the more extensively their DNAs will hybridize. The another one is polymerase chain reactions and then site-directed mutagenesis are the two waves by the DNA hybridization via renaturation is obtained by modern molecular genetic techniques. Non-enzymatic transformation, that is spontaneous mutagenesis, that is the changes in the amine group, which is referred as deamination, which is very slow reaction which has a large number of residues. The net effect is significant, 100 C2U events per day in a mammalian cells. The another by depurination, that is the purine is replaced by the another purine. N glycosidic bond is hydrolyzed and then significant for purines, 1000 purines lost per day in a mammalian cell. Fortunately, cells have mechanisms to correct most of this modification. The last one is radiation induced mutagenesis, that is mutations is induced by the different type of rays. First one is UV. UV light induces demineralization of pyrimidine, which cause kinks in DNA structure. This effect is the main mechanism of skin cancers. UV melting, that is ultraviolet melting, is a heat double-stranded nucleic acids causes the strands to unwind by disturbing the ordered stacking of the bases and breaking hydrogen bonds. The process can be conventionally monitored by an increase in the ultraviolet absorption as the double-stranded unwinds to single strand. The thermal denaturation of double-stranded DNA is progressively, and the concerned melting of the whole structure occurs at a well-defined temperature corresponding to midpoint of the smooth transition. This temperature is known as the melting temperature. Slow heating of double-stranded DNA causes the unwinding of the ordered helical structure into two single-stranded constitution. This has been seen as a sigmoidal curve of increasing UV absorption. The midpoint corresponding to the precious melting temperature of the duplex is indicated. To determine thermodynamic parameters, samples are made up of different concentration, each containing equal amounts of two complementary sequences of DNA in a salt or buffer solution. On mixing it ambient temperature, the two strands of DNA spontaneously form a duplex. This is true for DNA strands of any reasonable length. Each sample is heated in a UV spectrometer from 20 degree to 80 degree at a rate of 1 degree per minute, and the absorption is monitored. The melting temperature Tm is the temperature at the maximum absorption gradient and corresponds to 50% association. By using the calorimetry is a classically important method for measuring DNA duplex stability. Although UV melting has proved more popular owing to the available of UV spectrometers with the temperature controlled sample chambers and are relatively ease of recording temperature value, whereas UV melting point provides an indirect method for measuring delta H and delta S and delta G for melting. Calorimetry allows more direct measurement of these thermodynamic parameters. Apart from the UV rays, the ionizing radiation, that is X-rays and gamma rays, cause ring opening and strand breaking. Cells can repair some of this modification, but others cause permanent and determinal mutation. Accumulation of this type of mutations is linked to aging and carcinogenesis. How to overcome or tuning the DNA duplex stability? When designing and synthesizing synthetic DNA oligonucleotides, for example, for use in the antisense therapy or as a PCR probes, it is often described to either increase or decrease the stability of the duplex formed on binding to complementary DNA. Nucleic acid analogs. Artificial nucleic acid analogs binds to complementary DNA. Peptide nucleic acid, a neutral molecule, forms particularly strong and specific interactions with the DNA. Structures of peptide nucleic acid can be incorporated into the DNA oligonucleotides to increase duplex stability, but care must be taken in designing the PNA-DNA junction, and the stabilizing effect of the PNA can be offset by destabilizing impact of the junction. Locked nucleic acid and unlocked nucleic acid are analogs of RNA that can be easily incorporated into DNA oligonucleotides during solid phase oligonucleotide synthesis and respectively increase and decrease duplex stability. The locked and unlocked nucleotide phosphometric monomers are not readily available. However, it is not always easy to predict the effect of incorporating locked and unlocked units into DNA oligonucleotides before they are synthesized. Modified bases. Modified DNA bases that increase the stability of base pairs have been developed. These modified bases can be incorporated into oligonucleotide during solid phase synthesis and offer a more predictable method of increasing DNA duplex stability. APDCG clamp. APDCG clamp is a tricyclic analog of cytosine. Like cytosine, G clamps forms base pairs with guanine, but while guanine base pairs with cytosines are stabilized by 3 hydrogen bonds, GC clamp base pairs have 4 hydrogen bonds. The extra hydrogen bond can lead to an increase in temperature point of 18 degrees centigrade. The incorporation of the multiple G clamp bases can result in further increase in the duplex stability. 2-aminoadenine. 2-aminoadenine forms base pairs with thymine that are stabilized by 3 hydrogen bonds, more than 2 hydrogen bonds of 8 adenine and thymine base pairs. Each substitution of an adenine and thymine base pair by a 2-aminoadenine thymine base pair increase the stability of the duplex by about 3 degrees centigrade. Other modified bases are modest increase in duplex stability have been observed with the other modified bases including 5-methyl cytosine and propenyl cytosine and propenyl uracil. The stability of the DNA can be determined by various forces such as electrostatic repulsion, hydrophobic forces, hydrogen bonds, stacking interactions. In DNA structure, the base pairs are found in the interior of the helix and are charged, and hydrophobic sugar phosphate backbone are on the exterior. The phosphates from the two strands are also kept away from each other to reduce the electrostatic repulsion, somewhat neutralized by divalent cations like magnesium 2+ and polyamines. If the double-stranded DNA are heated, the chain separated and become random coils. This is referred to as denaturation. It is easy to monitor denaturation of DNA by measuring changes in absorption of light at 260 nanometer, the wavelength at which the bases absorb maximally. On denaturation, the absorption of a solution of DNA increases by 20 to 30%. This is called the hyperchromic effect. The effect has its origin in the stacking of the bases in the double helical structure. The absorption maximum at 260 nanometer originated from the pi electron system of their rings. When the bases are stacked, interaction between the pi electrons of the bases leads to a coupling of their transition and decrease the absorption. When the bases are unstacked, this effect is removed. The type of denaturation curve obtaining the temperature at which the increase in absorbance reaches 50% of its maximum is called the melting temperature. For the two cases The melting temperature value depends on the nature of the solvent and is lower at lower ionic strength. If the solvent conditions are kept constant, then the most important factor governing melting temperature is the base composition of the DNA. The higher the content of G and C, that is guanine and cytosine, the higher in the melting temperature. This is as expected because the guanine and cytosine base pair makes 3 hydrogen bonds, whereas adenine and thymine pair makes only 2. Indeed, there is an empirical quantitative relationship between the percentage, that is G plus C content, and the melting temperature are as follow as percentage of guanine plus cytosine is equal to 2.44 of melting temperature minus K, where K is a constant which depends on the solvent, can be used to determine the percentage of G plus C for a newly isolated DNA molecule. Stacking interaction stabilizes the double helix about as much as base pair of hydrogen bonds. Stacking interaction are not sufficient to overcome the electrostatic repulsions of the phosphate. The hydrogen bonding guarantees the complementary of the strands. The hydrogen bonds in the minor groove formed due to the formation of water spine. DNA can be denatured under extreme conditions of temperature or pH. Condition disturb the hydrogen bonding and hydrophobic interaction between the bases and result in separation of the strands. Denatured DNA is less viscous than native double helical DNA and the base exhibit greater UV absorption. The transition from double stranded DNA to single stranded random coil DNA is called a helix coil transition. Hydrolysis of acid and alkali. DNA is generally quite stable, resist attack in acid and alkali solution. In mild acid solution at pH 4, the beta glycosidic bonds to the purine bases are hydrolyzed. Protonation of purine bases, that is N7 of guanine and N3 of adenine, occurs at this pH. Protonated purines are good leaving groups. The depurinated sugar can easily isomerize into the open chain form, and in the form depurinated DNA is susceptible to cleavage by hydrolysis hydroxyl ions, whereas RNA is very unstable in alkali solution due to hydrolysis of the phosphodiester backbone. The 2OH group in the ribonucleotides renders RNA molecule susceptible to strong cleavage in alkaline solution. There are many enzymes that cleave RNA ribonucleases. In addition, there are some of the environmental factors which affect the helical stability. First one is salt concentration. The effect of salts on oligonucleotides at salt concentration above 1 molarity, addition of salts lowers the melting temperature of DNA. The lowering is relatively dependent on cation but is strongly dependent on anion. The better denaturants are most effective in enhancing base solubility. Second one is solvent effects. Addition of co-solvents to aqueous solutions of nucleic acid typically destabilize the ordered form, and this is often used as a substitute for denaturation by temperature. The effects of co-solvents have been studied by measuring the concentration of co-solvent required to give 50% denaturation of T4 DNA at 73 degrees centigrade, and by measuring the effect of co-solvents on thermal denaturation curves of various oligonucleotides. The third one is pH. The stability of most double helix is relatively insensitivity to pH between 5 and 9. At lower and higher pH values, stability is decreased. At low pH, bases in the single strand bind more protons than in the duplex, thus favoring single strand. At high pH, guanine, thymine and uracil are deprotonated, thus precluding normal hydrogen bonding and increase charge repulsion. These are the few things about the stability of the nucleic acids and how the stability is disturbed by the various factors such as physical and chemical factors. By maintaining these factors appropriate to the stability of the DNA, we can able to stabilize the nucleic acid at various conditions. Thus, we concluded this episode and we will discuss a new topic, prokaryotic and eukaryotic genome organization, in the next episode. Thank you.